So we know that cancer patients who receive chemotherapy or radiation therapy are at higher risks of developing myeloid neoplasms. And traditionally, we actually thought that this was due to therapy causing mutations that eventually lead to some people developing myeloid neoplasms. But in 2014, a really seminal paper came out from Dan Link and Terrace Wong and others that showed that, interestingly, among solid tumor patients who later developed therapy-related myeloid neoplasms, you could actually detect some of the mutations that led to the neoplasm even before they had been exposed to any chemotherapy. So it seemed like in that case, chemotherapy led to the expansion of mutations and the acquisition of additional mutations that led to cancer. At the same time, there's been several large sequencing studies that show that it's normal and probably just a process of aging that our hematopoietic stem cells develop mutations, and that leads to their clonal expansion over time. And this is what we refer to as clonal hematopoiesis. In the general population and in solid tumor patients, we know that clonal hematopoiesis leads to higher rates of leukemia. And we also know that solid tumor patients who have been exposed to oncologic therapy before have slightly higher frequencies of clonal hematopoiesis. And so to understand this relationship between clonal hematopoiesis and oncologic therapy, we assembled a large cohort of solid tumor patients at MSK. So among these 20,000 solid tumor patients at MSK, they had their blood sequenced actually as a matched control for solid tumor sequencing. And their blood was sequenced using a targeted panel of known cancer driver genes at a mean coverage of around 500x in the blood. So this enabled us to actually call clonal hematopoiesis in all of these patients. So we saw across most ages that, that CH was present overall in about 30% of solid tumor patients, and that across most age ranges, the treated people were slightly more likely to have clonal hematopoiesis than untreated people. And as others have shown, this appeared to be especially enriched in 3 specific genes, PPM1D, TP53, and CHEK2. These are all genes that are involved in the DNA damage response pathway, as you know, and all seem to be the most greatly enriched in the blood of patients who had been exposed to oncologic therapy. So we wanted to next understand whether CH was related to some regimens more than others, which is an important clinical and biologic question. So we looked at how specific therapies were associated with CH in this large cohort, and found that indeed CH was associated with, more specifically, with radiation therapy, with cytotoxic therapy, and even among cytotoxic therapy regimens, it seems like topoisomerase II inhibitors and platinum-based regimens had the strongest association with clonal hematopoiesis. And even among the platinum-based agents, carboplatin, which is probably the most myelotoxic platinum-based regimen, showed the strongest association with CH. And again, looking at these specific regimens, this seemed to be the strongest for mutations in the DNA damage response pathway. And so next, we wanted to answer a fundamental question about whether treatment was promoting pre-existing CH or inducing new mutations. So to answer this, we collected a second blood sample in over 500 patients. And we found that among these 2 samples, mutations were present almost always at both time points. And then indeed, among patients who were exposed to cytotoxic therapy or radiation therapy during the follow-up period, it was these patients where we saw the most growth of CH mutations. And again, this appeared to be very specific to mutations in the DNA damage response pathway, which is, again, PPM1D, TP53, and CHEK2. And so finally, getting at the question that was initially posed to me about the relationship between clonal hematopoiesis and progression to therapy-related myeloid neoplasm, we assembled data from MSK, where we had 35 patients who eventually developed therapy-related myeloid disease, and combined this with 3 other published data sources for the largest combined cohort for therapy-related myeloid neoplasm risk. And so using this combined data set, we show that a combination of blood count parameters and CH-specific mutational features can define patients who are at higher risks of developing therapy-related myeloid neoplasm. And as you can see, actually, the hazard ratios for some of these risk factors are quite high. For example, patients who had 3 or more mutations had hazard ratios of over 20 for developing therapy-related myeloid disease. But as you know, therapy-related myeloid disease is a very rare occurrence. So for example, for women with breast cancer, even though they have higher relative risk than the general population, their 10-year absolute risk of therapy-related myeloid disease is only 0.3%. So in order to understand whether this might have clinical actionability, you really need to look at the absolute risk of therapy-related myeloid disease. And so we did this in breast cancer using a synthetic model and showed that for women at the top 1% of risk based on their CH molecular profiles, that when we modeled giving or not giving adjuvant therapy, we found that if one were to defer adjuvant therapy in women in the top 1% of risk, that it could reduce the absolute risk of therapy-related myeloid disease by over 9%. And this is more than the benefit that most women will get in terms of overall survival benefit from adjuvant therapy for breast cancer. This is just a model meant to demonstrate that risk stratification of AML and MDS in solid tumor patients could have clinical actionability. And ultimately, this will need to be validated in cancer-specific settings in order to actually bring this into clinical action. So in summary, we show that clonal hematopoiesis is shaped by environmental exposures in a gene-specific manner. We showed that platinum, topoisomerase II inhibitors, and radiation seem to have the strongest selective pressure for CH, specifically in the DDR pathway, mutations like TP53 and PPM1D. And that therapy-related myeloid disease prediction of this using clonal hematopoiesis could inform treatment decisions. And we hope that this will spur further research to actually bring this into clinical action. Thank you.